Okay, so this video is going to be about the M phase, which is 10% of your cell's life, and it's actually composed of two main parts, mitosis and then cytokinesis. And mitosis has actually 5 stages. We have our prophase, prometaphase, which is an intermediary stage between prophase and then metaphase. Then we're going to have anaphase, and then we'll have telophase. After that, we have cytokinesis, and cytokinesis is division of the cytoplasm. So mitosis, its definition is division of the nucleus, and that's huge. It's not cell division, it's division of the nucleus, because mitosis is going to divide the nucleus, which is all your chromosomes and DNA. And it's going to make sure that that is done accurately first. And then once that is done, then the cytoplasm divides. And the cytoplasm is the cytosol plus the organelles. Okay, so first we got to divide up the big stuff, the chromosomes, and then after that we can separate the cytosol and the organelles. All right, in order to do this drawing, I'm going to have you kind of do a little bit of an art project. I want you to take 2 sheets of paper, and I want you to tri-fold them so that they have somewhat equal sections, or you can measure it with a ruler, whatever, so that there's 3 separate sections here and 3 separate sections here. And then down that crease, you're going to draw a line on both pages. Okay, and then take it and fold it in half. Oops. Nice and neat for all you little OCD kids who like everything to be nice and neat. So it'll be right down the middle. So then you have 6 equal blocks on one page. Yeah, I know mine's not very nice and neat, and that'll freak some of you out, but you'll be fine. And then 6 equal boxes on another page. Okay, and you can print it out if you want to, if that'll make you happy. But in the end, what we're going to do is I'm going to turn it over, and I'm going to tape it together on the back, because I'm going to be writing all over the front, and it's hard to write over tape. And so your finished product, and I'll trace down this line, will look something like this. Okay, so you have 1, 2, 3, 4, 5, 6 boxes. Okay, so you want to have 6 boxes, and that's what I'll be drawing in. I'll be drawing some images on the top, and then I'll be writing some information on the bottom. Okay, so now you should have your 6 squares on top, 6 squares on bottom, right? And in the first box, I just want you to cut it in half, okay, because we're going to put a little information in there. So at the top of the first box, I want you to put interphase, okay? And then along this line, I want you to write cell signaled, okay? So this is where between interphase and our first step here, the cell has to get signaled, and it's going to transition from interphase into prophase. Okay, so our first step of mitosis is going to be prophase. So this is mitosis. All right, our second step is going to be called prometaphase. And it's just a little in between. This is also mitosis. All right, and then we're going to go to metaphase, which is also mitosis. And then we're going to go to anaphase, which is also mitosis. And then we have telophase. And this is the end of mitosis. Okay, and the last box is for the next thing that occurs, and the last thing that occurs is cytokinesis. Okay, so there's cytokinesis at the end. It is not part of mitosis. Okay, so mitosis, you learned was 4 steps. In AP Bio, we get a little more specific and we make it into 5 steps. So it's P and then M, so there's PM together, and then A, T. So it's still PMAT, but we have a little intermediary stage here, okay? So we'll be drawing pictures up top, and then we'll be writing information and whatnot at the bottom. So when a cell is in interphase, that's when it's working and it's doing its job. So if you were to look at it under a microscope, it would have a dark spot inside the nucleus, and then the nucleus would also be shaded in. Okay, so this is our nucleus, and this dark spot is called the nucleolus. And the nucleolus is the organelle that makes ribosomes. So the cell is about to stop working. So it gets the signal in G1, and then it'll go to the S phase, and then it goes to G2. And then after G2, it hits that prophase, okay? So interphase is 90% of the cell's life. And then this whole mitosis, cytokinesis is only 10%. All right, so let's see. We would say the interphase is the longest phase of a cell's life. Remembering that some cells stay in there forever. Some cells stay in there for 20 years. Some cells prefer to stay in interphase, but because of damage and different things that we do, they have to enter mitosis faster than they want to. Some cells can be in interphase for as short as 3 days. It just really depends on what type of cell we're talking about and where it is. Okay, so with interphase being that longest phase, G1 is right after it's created. The S phase has to happen. S phase must happen. So that there is an extra copy of each sister chromatid. Okay, so remember those, there you go. Those sister chromatids are identical, and a cell that is in G1 only needs one sister, so it doesn't really actually need to replicate, make a copy, because it's not planning on dividing. But once it gets that signal and it finds out that it has to divide, it will go through this S phase and replicate and make a double copy. So now instead of a single chromosome like this, it doubles like this, and then they stick together so they don't lose each other. And then they're able to divide and split up later on in one of these phases of mitosis, okay? So now we're gonna go into the actual phases of mitosis. So prophase is the first phase. Pro means first. And so in prophase, the signal has been given. All right, so a couple things are gonna happen. I'm actually gonna do each phase on a bigger sheet of paper so that you can see everything. You do yours in the box, but I'll do mine on the paper. Ooh, that's very blurry. So this is going to be prophase. Okay, so this is your first phase. So if you were to look at a cell in prophase, some of the things that are occurring is, I'll explain this after I write it. Chromatin condenses and forms chromosomes. Okay, so if you backtrack and you were to look at a cell in interphase, okay, you can't see inside the nucleus because the nucleus is dark. But if you could see inside the nucleus, you would see the nucleolus, and then all the DNA is actually unraveled. So one chromosome, as we know it, glued together, this piece is a sister chromatid, and I told you it's a whole long strand of DNA. So if I was to start pulling it, like if it was a sweater, and unraveling it, it's literally just one long, huge strand. So I could just pull it and unravel this whole sister chromatid, okay? So this is one long strand. And while the cell is working in the interphase, that DNA is unwound, so it actually can read the directions and understand what's going on. So this is a working cell, okay? When we go into that prophase, the cell has to pack up that DNA, and so the nucleus is going to start to disappear. So that's another little bullet. The nuclear envelope begins breaking down, because the whole premise of your mitosis is division of the nucleus. So we got to get to these chromosomes. So this, I mean, if this was a separation, this would be the money. This would be the big stuff, okay? So the chromatin is going to condense. The envelope around the nucleus is going to start breaking down. And when the chromatin condenses, chromatin is DNA plus proteins. So DNA is like wrapped around these proteins called histones, and it just keeps the DNA from getting tangled up. So when you talk about the DNA wrapped around proteins, it's called chromatin. So when it condenses means to bunch together. So where in a working cell, the DNA is all unraveled, in a dividing cell, the DNA condenses and it bunches back up into these structures that we call chromosomes. So microscopically, it's just a long bunched up strand of DNA. Another thing that occurs is the nucleolus disappears. Okay, so when a cell is not working, it does not need to make ribosomes, because ribosomes are making proteins, so that's not necessary. And then all of a sudden, what is going to start showing up are these guys. Okay, now every artist draws them a little bit different. I'm gonna draw them looking like little trash cans or whatever. Some of them draw them and they're like shining. Okay, so these organelles are called centrioles. Okay, and the centrioles are in charge of the cell division, right? I'm gonna put a little star by this. This is only in animal cells. Okay, so you will not find centrioles in plant cells. So plant cells sometimes do not divide like they should, and that's probably a consequence of this. And then these little things coming out of them, okay, are spindle fibers. And spindle fibers are protein fibers, very similar to muscle fibers. So they're very stretchy and flexible, but they're strong. And so they are going to help move these chromosomes around and get them lined up like they should. So prophase, we got chromatin condensing and forming chromosomes. The nuclear envelope is beginning to break down. Begins breaking down. I did not finish that. Excuse me. The nucleolus disappears. The centrioles appear. Okay. And we are in our first phases of what's going on. Now, if this was a human cell, there would be 46 of these chromosomes, okay? I'm just going to draw 4. So I've got 1, 2, 3, and 4, okay? It's always an even number because the chromosomes are always in pairs. Now, it can be an odd number of pairs, but it's always an even number of chromosomes, okay? So there's that cell in prophase, and then it's going to transition into its next phase, which is prometaphase. Okay, and then I'm just going to draw metaphase right beside it. So that's what your paper looks like. So you're good to go on that. Okay, so in prometaphase, is it just a transition? So I'm going to skip that for a second. I'm going to go to metaphase. Okay, metaphase, little hint word to help you, is called middle. All right, so in metaphase, it's when your chromosomes are going to line up in the middle, and the middle of the cell is kind of like what we call the middle of the earth. So if I was to draw a dotted line down the earth, the middle, I would call it the equator. Now, depending on how you're looking at a cell, the middle can be this way, this way, this way, whatever. So some books draw it one way, some books draw it another way. It doesn't matter. In your book, they have them drawn left to right across, but so they draw the equator like the earth equator. But then a lot of people draw them up and down. It's the same concept. So we'll do prometaphase in the middle in a minute. But at the end of metaphase, what you should have is your 4 chromosomes lined up straight down the equator. Now, I want you to imagine that they're like kindergartners. Kindergartners are not the best at lining up. So you need to assist them and help them. So what has happened is over here in prometaphase, these centrioles have moved, and they're moving to the sides of the cell, and they're getting in position, and they're going to end up being right here. Okay, so if this is the equator, what would the top and the bottom, so if I turn this sideways, if this was the earth and this was the equator, what would we call the top and the bottom? Hopefully you know that we call it the poles. Okay, so in prometaphase, the centrioles begin to move to the poles. Okay, so the poles are just going to be opposite of the equator. In metaphase, the centrioles are at the poles. All right, so coming out of the centrioles are these spindle fibers. Okay, and they're going to get longer, and they are going to, spindle fibers attach to the chromosomes. Okay, just like if you were trying to line kids up, so they're going to attach these chromosomes and start moving them to the equator. Okay, so in metaphase, we have them all lined up. Now notice my spindle fibers have not let them go, okay? So I've got 2 attached to every one of these chromosomes, but there's probably hundreds of them. And this is going to be like little elastic fibers. And so these guys are going to be trying to move all these chromosomes and get them to line up like they're supposed to. So yeah, good luck to them. So they're making all these chromosomes move and line up to where they are finally in metaphase lined up in the middle. So if you were to look at your images, okay, so prophase, they just become visible and the centrioles show up. Then the centrioles start moving until they get to the poles. But as they're moving, they send out their spindle fibers and they start attaching to these chromosomes. And then they're moving these chromosomes into position where they are all lined up. Okay? Super important, because if we don't get them all lined up, then we're going to have an issue with our division. Now, these fibers are strong and flexible. All right, and so basically what's going to happen right now is during metaphase, the two centrioles are playing tug of war. Okay, so what the cell would look like is it actually starts getting more shaped like a watermelon, because this centriole starts pulling as hard as he can towards his pole, and then this centriole starts pulling that way. So what they're doing is they're getting as much tension on those fibers as they can, because they're going to tug and tug and tug. Now, from the first video, I believe, I showed you that a chromosome is two sister chromatids held together in the center by a protein disc. Okay, so this protein disc has a very important role. It is keeping these sisters together so that the centrioles can get everybody lined up. And as soon as they get lined up, we're going to actually rip these sisters in half, which is kind of sounds terrible, but you want to make sure that completely half of each pair of sister chromatids, you want a whole piece to go to the left, and you want the other complete entire sister chromatid to go to the right, because then that ensures that the new cell that is being formed over here, its nucleus gets a full sister chromatid. Same thing here. You're going to hope that these spindle fibers get half of that chromosome and half of that chromosome, so that now they each have a complete full sister chromatid, because all you need is one sister chromatid. And if you have one sister chromatid, then you have exactly what you need for all the directions, okay? So this is the longest phase of mitosis. Not the longest phase of the cell, the longest phase of mitosis, because you have to make sure you have everybody in place and everything is exactly how it should be, because when you rip them in half, if you rip them wrong, then one cell gets too much DNA and one cell doesn't have enough, and it's not fair. All right, so the next phase is A, and A stands for anaphase. So I'm going to do anaphase and telophase together, side by side. So here's anaphase, okay? And then we'll have telophase. And then I'll finish up with cytokinesis. Okay, so now, your picture, you have your metaphase here. So they're all lined up. So A stands for away. So if you're looking at cells and you see that the chromosomes are still intact, you are still in metaphase. Once they rip in half, you are now in anaphase. In anaphase, the chemical reaction occurs. There's a lot of things that go on there, and if you've studied biochemistry, you'll learn a lot about that. But a chemical reaction occurs that results in, I would say, an enzyme. I would say the breakdown. Okay, an enzyme is going to break down the centromere. Okay, and the centromere is that protein disc that holds these sister chromatids together. So as soon as that occurs, right, I want you to imagine you're in tug of war and somebody comes along and just literally cuts the rope in half. Everybody's going to fall backwards, because this group is pulling this way and this group is pulling that way. So if we're in a tug of war, we got our little people over here and they're all hanging onto their rope. And over here, you got your people trying to hang on to this rope, and everybody's pulling in their directions. And then I'm going to come along and I'm just going to cut the rope in half. Okay, this group's falling down and this group's falling down. And the reason it's done that way is so that these sister chromatids don't get stuck in the middle. They are pulled so tightly in the two directions that they shoot backwards and go rapidly to their side. So this one and this one. So he's going to go that way because we just ripped them in half. And remember, they're attached by spindle fibers. So these spindle fibers are going to pull them towards their pole. So second one gets ripped in half, third one gets ripped in half, fourth one gets ripped in half, okay? With spindle fibers pulling them all back towards the pole, okay? And rapidly, all right? So this is anaphase, where the sister chromatids are ripped apart and they're pulled away from each other towards their centrioles. All right, and then the last and final phase of mitosis and cell division is telophase. And I always say telophase stands for two new nuclei, because mitosis is division of one nucleus into two nuclei. So I'm going to start drawing it a little bit pinched in. So it's going to look kind of like a little butt. Because what's going to happen is these little pieces of the membrane are going to start pinching in a little bit because there's these little bands of proteins that are starting to wrap around it. And when we did that bubble lab, I had you draw a big bubble or blow a big bubble, and then I had you like take string and pull it through. And that was to try and kind of create a separation here. That's what's going to happen in the last phase in our cytokinesis. But in telophase, what's going to happen is around each new cluster of sister chromatids, we're going to start trying to form two new nuclei. Okay, because what we want to do is we want to wrap them up tight so that they don't join back together. Okay, so two new nuclei begin to form. Change my marker, that one's getting dry. The centrioles begin to disappear. They're not needed anymore, so they're going to go away, and they'll wait until that cell decides it needs to divide again. The chromosomes become less condensed. So what that means is they start spreading out a little bit. All of the cytosol and everything, you've got organelles, and they're going to make sure they all kind of separate and spread so that we have a little bit of everything, all right? And then this pinching in is called cleavage. All right, and that actual location is a cleavage furrow. And a cleavage furrow is just where that that cell membrane is starting to pinch in, because it's going to try and separate into two completely new cells. All right, so the last thing that you're going to have is cytokinesis. So that'll be your last box. Okay, and cytokinesis means splitting of the cytoplasm. And remember, cytoplasm is the organelles plus the cytosol. So now what would end up happening is that membrane pinches in, and it keeps pinching in and pinching in and pinching in until all of a sudden it touches. And then now we have two completely separate cells. So once it has completely separated the cytoplasm, you have completely two new cells. This is cytokinesis. This cell will have its own nucleus with 46 sisters, and this one will have its nucleus with 46 sisters. And these cells will directly go straight into G1, okay? And they'll start working. Now, a plant cell does all of this same process, except when it gets to cytokinesis, its membrane cannot pinch in because it has a wall around it. Okay, so this is animal cell and bacteria. Okay, they're going to do cytokinesis. But a plant cell, it has its new nuclei forming here and its new nuclei forming here, but it can't really pinch in anything. Okay, so its plasma will separate, and its cytosol will separate, but then it starts building a wall. Okay, and it builds a wall from the middle to the outside, and this middle wall is called a cell plate. Okay, so now with your animal cell, you have two new daughter cells that are identical, and with your plant cell, you have two new daughter cells that are identical. And eventually, the cell plate grows, you get a cell wall, and this will separate into two new plant cells. All right, so to summarize, we started with one with 46 chromosomes, and it went through prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. And at cytokinesis, we get two separate daughter cells with 46 chromosomes each. Now, this is a somatic cell, and these are also somatic cells, which stand for normal body cells. And they have all the chromosomes in them. So scientists call them 2N, right? So this is a 2N, and this is a 2N. They also refer to them as being diploid. Di means two. All right, and these are also diploid or diploid. People say it different ways. But that means that these cells have all the chromosomes. There's two of every chromosome in the cell. And we're going to learn later on that those two, one of each of the two came from mom, and one of each of the two came from dad. But this whole process, this is mitosis, and then plus, cytokinesis, and then we get two new cells. Okay, mitosis, division of the nucleus, cytokinesis, division of the cytoplasm, boom, two new daughter cells. And every single cell in your body does this at different times, but this is how new cells get made. Have a fabulous day, and our next video will be about cancer. We'll talk about how this process works, but it works when we don't want it to. And so you're going to have uncontrolled cell division, so uncontrolled mitosis. Goodbye.